Students asking ribosomes. Ribosomes are called as suicide bags. It is not a ribosome. Lysosomes. It is lysosomes. These are called as suicide bags. Ribosomes are not called as suicide bags. They are the lysosomes. Okay, these having a proteolytic enzymes, and these enzymes are hydrolytic enzymes digest the various components of the cell. Hence, the lysosomes are called as suicide bags. Okay, so these are not suicide bags. Then one more student asking the difference between light reaction and dark reaction. Light reaction, dark reaction. So we'll learn this concept in detail in photosynthesis topic. Here we can discuss in short. Light reaction means light harvesting reaction. Light harvesting reaction. Here solar energy is trapped. Is trapped. Dark reaction is that assimilate the carbon dioxide and synthesize the glucose. Dark reaction absorb the carbon dioxide and synthesize the glucose. So this one light reaction harvest the solar energy and split the water. Light reaction split the water. Dark reaction absorb the carbon dioxide and synthesize the glucose. So it is a short in detailed concept we will learn in the next topic called as photosynthesis. ATS ribosome and 70S ribosomes. Difference between these two is these having more sedimentation coefficient. Here it is a Swedenborg's test tube. Swedenborg's density gradient test tube. Swedenborg's density gradient test tube. When he centrifuged different ribosomes, based on their size, based on their molecular weight, based on their density, they lie in different positions. Here it is 80 position, 70, 60, 50, 40, 30 like that. Okay, whenever this ribosome is locating at the ATS means it is ATS. Here it is 70 years, it is 60, it is 50 like that. Based on their size, based on their molecular weight, they settle at the different position in the Swedenborg's density gradient test tube. So ATS having more molecular weight, it is having more density, it having larger size, it is a larger size than the 70s. But they are present in different organelles and different cells. About tRNA, nothing but transfer RNA. This tRNA meant for carrying the amino acids to the sites of protein synthesis. We learned in second year what are tRNA. These are the RNAs carry the amino acids to the site of protein synthesis. So it is called as tRNA. One more student asking about lumen. Here we discussed about the thylakoids. Thylakoids are the structural and functional units of the chloroplast where we find a light reaction. So these thylakoids having a membrane, and this cavity of the thylakoids is called as lumen. The cavity of thylakoids are the portion at the center of the thylakoids is called as lumen. Okay, present in the thylakoids of the grana of chloroplast. The student asking how does the chromoplasts are formed by the chloroplast. Usually, chloroplast having the chlorophyll pigments. Okay, but during senescence phase of the leaves or some parts of the plant, the chloroplast lose the chlorophyll pigments, and these chlorophyll pigments, when disappeared from the chloroplast, they convert into chromoplast because of remaining pigments are only carotenoids or phycobilins or phycoerythrins. Okay, that's why they convert into chromoplast. Chloroplast lose the chlorophyll pigments and form a chromoplast.
ये ना one of the students asked a doubt about what is the function of the stromal lamellae. Two grana are interconnected by stroma lamellae. This is called as the stroma lamellae, and this stroma lamellae it transfer photosynthetic pigments from one grana to the another grana. You know very well. So photosynthetic pigments which are present in the grana. So individual structure is called as thylakoid, which are arranged one above the other like a pile of coins. So these contain the photosynthetic pigments, and these photosynthetic pigments are transferred from one grana to the other grana through this stroma lamellae. So this is the function of the stroma lamellae.